Hello, my name is Kenneth Boote. I'm at the University of Florida and co-authors of Tceiling and Garrett Hogenboom helped with the programming, and Prasad helped with some early work with the peanut grow aflatoxin model. Aflatoxin is a serious problem in much of the world, particularly in developing countries, and aflatoxin contamination of the seed poses a continuing health risk to consumers. Infection by the Aspergillus fungus and aflatoxin synthesis are increased under conditions of drought and elevated soil temperature, which are prevalent under rainfed agriculture. The objective of this presentation is to illustrate the addition of capability to the CROPGRO-Peanut model to simulate infection of pods by Aspergillus fungus and the subsequent synthesis of aflatoxin in peanut seed. I'll give a little bit of history of the CROPGRO model, which is in the DSAT system. CROPGRO-Peanut itself was developed in the mid-1980s. It's part of the DSAT. I participated during 2002 to 2004 with the Southeast Peanut Grower Initiative on aflatoxin. Prasad was a postdoc with me then, and the two of us coded aflatoxin prediction in the CROPGRO-Peanut model. That code was basically lying dormant, almost lost. And then in 2018, the International Food Policy Research Institute contacted me and indicated an interest in simulating aflatoxin contamination in peanut with the CROPGRO-Peanut model and doing the same with cereus maize. This project was funded to me and to Garrett Hogenboom, and we used data from ICRISAT. It's a published paper with Waliyar et al. 2003. Just a little bit about the CROPGRO-Peanut model. Its mechanistic model runs with a daily time step. Some things run hourly, and it simulates carbon, nitrogen, water balance of the crop, and on a daily basis adds pods and grows the pods and the seeds within them. And there are successive pod cohorts, and we basically modified the code so that we kept a record of the soil temperature and soil water status at the 5-centimeter depth, along with plant water status. Just a little bit about the cohort structure. Now, peanut will flower over a 30 to 40 day period, and each day's group of flowers forms after a time period that's called FLSH. A pod begins to grow from that flower, peg, and then a flower, then a pod, and there's a shell growth duration, and then after a later time the seeds begin within that same pod. There are multiple cohorts layered that may continue over 30 to 40 days. The goal here is to track the soil temperature, soil water status, and plant water status during that applies to each of these cohorts, and then predict infection and synthesis of aflatoxin. Some of the principles involved with the coding, and it is based on the daily addition of pod and seed cohorts. The infection process is based on soil water, the plant water status, which is SWFAC, and soil temperature. And NPP basically is a counter for the different cohorts. This may number from 1 to 40 or so. So this is the infected number of pods is equal to the infected number of pods, and these are the uninfected ones, times a rate constant. This is the soil water status in the 5-centimeter root zone or pouting zone. Plant water status is 1 minus SWFAC, and this is a temperature modifier. The next step in this is to create aflatoxin synthesis in those infected pods. Okay, and this is done as a function of soil water or the plant water status and the soil temperature. It does not use the soil water status in the potting zone. And plant water status was more important in our calibration. Basically, the AF mass is the mass of actual aflatoxin, and then this creates the growth of aflatoxin based upon infected pods, a rate constant, plant water status, and a temperature modifier. The model outputs percent infected pods, so this is a summation in the model. So we get a infected pods and summing over all cohorts, and percent infection is infected pods divided by total pod number. The actual mass of aflatoxin is summed over all the pods, and we actually calculate concentration, part per billion of the seed as the mass of aflatoxin divided by the weight of total seeds. This illustrates the temperature function. The function for infection is this quadratic function, the blue, with a T base of 22, T optimal between 32 and 35, and a failure point at 45 degrees Celsius. This is based on soil temperature. This is not shown as a quadratic, but it is. But in synthesis has a base between 26, optimum at 28 to 30, and decline to 38 degrees Celsius. This illustrates the success after model calibration. We did modify some of the temperature functions, and they don't agree exactly with the ones from Dawson, Georgia. I'm not sure why. But simulated aflatoxin compared to observed, and the blue line is a regression with the slope 0.97. This is if we have an intercept in the line, but we had pretty good predictions. This is with Sadore, 3 years of data. Percent infection, in fact, was quite high in most of the observed. I'm not sure whether they may not have had some insect problems and other things with this cultivar, JL24. So there is some relationship of some predictability there, but it's not as good. This illustrates the prediction of pod number, blue, versus time, and the grain mass starting after that. And the green shows the water stress simulated by the crop model, and the black line is indicating infected pods, and then the blue illustrates aflatoxin increase part per billion over time for Sadore in 1994. A little bit of simulation illustrates for 12 seasons at Tifton, Georgia, roughly 1990 through 2002, and illustrates that one out of the 12 years had greater than 20 part per billion, so quite disastrous, but 10 out of the 12 falling below the 2 part per billion trigger. Basically, if you had high yield, basically you had not much aflatoxin, so there was a strong relationship of aflatoxin occurring under low yield conditions caused by drought. The conclusion that I would come to for this presentation, we have the code saved on GitHub. IFPRI is using it. We plan to incorporate this capability into a future release of the DSAT. We use the same functions for both peanut and maize later on. Soil for peanut, use the soil temperature. For maize, we use air temperature as a driver. The infection process has this base temperature with a quadratic function of between 22 to 32, 32 to 35 is the optimum. Aflatoxin has a very sharp function. It starts at 26 degrees Celsius and increases to a maximum between 28 and 30. I should point out that the temperatures, soil temperatures simulated by the model for Sadore Mali, hot dry season, range from 26 to 36 degrees Celsius. So it's right in this range. What we learned is that accurate simulation of crop water deficit is important, and it is more important than actually temperature. APRES is doing regional predictions with this same model.